Today, the team one, conformed by Samantha Hernández, María Alondra Delgado, Carolina Ramírez, Emiliano Ledezma, Gabriela Vázquez, and your server Anhuar Marroquin, we are going to present the comprehension activity of stage 2.
First, we are presenting the photosynthesis. The photosynthesis or chlorophyll function is the conversion of the inorganic matter to organic matter thanks to the energy provided by sunlight. In this process, light energy is transformed into stable chemical energy, with NADPH and ATP being the first molecules in which this chemical energy is stored.
I will present the cellular respiration. It is a set of chemical reactions by which certain organic compounds are degraded by oxidation to become molecules, which in the process generate energy that the cell can use for various functions.
Now I'm going to talk about fermentation. Fermentation is a catabolic process by which microorganisms obtain energy from organic compounds such as sugars and can transform them into simple chemical compounds such as carbon dioxide, acids, alcohols, among others. Fermentation involves using microorganisms to transform organic matter catalyzed by enzymes. A food is considered fermented when one or more of its chemical components are affected by useful microorganisms, so its resulting chemical composition is modified. Although fermentation does not generate as much energy as aerobic respiration, it provides adequate life support for many organisms.
Then we are presenting the phase of photosynthesis. We are two, and the like-independent phase and the like-independent phase. The like-independent phase, in this phase, photosynthesis must be on like-dependent location. This phase takes the light energy absorbed by the pigments in the plant's chloroplast and stored in an adenosine triphosphate, ATP. And ATP molecules combines adenine, sugar with ribose, and triphosphate groups. ATP is a key ingredient in the final phase of photosynthesis, which occurs independently of light-independently phase, and in this phase is also called the dark phase, assuming that this takes place in the dark. However, this phase is not depends on sunlight, can take place anywhere and at any of the late or light phase. ATP that was stored during the previous phase combines with the carbon dioxide to form glucose. A byproduct of the reaction is oxygen, which is released through holes in the leaves called stomata.
Phases of cellular respiration. The pyruvic acid produced by glycolysis is transformed into CO2 and H2O, and in the process 30 to 32 ATP molecules are produced. To know this process, we are going to see step by step the 4 stages of cellular respiration.
Glycolysis. In glycolysis, glucose, a 6-carbon sugar, undergoes a series of chemical transformations. In the end, it turns into 2 molecules of pyruvate, a 3-carbon organic molecule. In these reactions, ATP is generated and ADA plus is converted to NADH.
Pyruvate oxidation. Its pyruvate from glycolysis travels to the mitochondrial matrix, which is the innermost compartment of the mitochondria. There, pyruvate becomes a 2-carbon molecules attached to coenzyme A, known as acetyl-CoA. In this process, carbon dioxide is released and NADH is obtained. Citric acid cycle. The acetyl-CoA obtained in this previous step combines with the 4-carbon molecules and goes through a cycle of reaction to finally regenerate the initial 4-carbon molecule. In this process, ATP, NADH, and FADH2 are generated, and carbon dioxide is released.
Oxidative phosphorylation. The NADH and 2-FADH produced in previous steps deposit their electrons in the electron transport, change and return to an empty, forms NADH plus and FAD. The movement of electrons down the chain releases energy that is used to pump protons out of the matrix and form a gradient. The protons flow back into the matrix through an enzyme called ATP cytase. to make ATP, and the end of the electron transport chains, oxygen receives the electrons and pick up protons from the medium of the water.
Alcoholic fermentation. In this process, NADH gives its electrons to a resultant of pyruvate and produces ethanol as a final product. This process is formed in two steps. In the first step, a carboxyl group is removed from the pyruvate and this is released as carbon dioxide. This causes acetaldehyde to be produced, which is a molecule composed of two carbons. While in the second step, NADH gives its electrons to acetaldehyde and this causes NAD+ to regenerate, producing ethanol at the same time. This process produces the ethanol in alcoholic beverages such as wine and beer.
Lactic fermentation. Lactic fermentation is an anaerobic cellular process where glucose is used for energy and where the waste product is lactic acid. This process is carried out by many bacteria called lactic acid bacteria, fungi, some protozoa, and in animal tissues. The process consists in converts 3-carbon pyruvate to 3-carbon lactic acid. The hydroxypyruphonic acid regenerates NAD in the process. Thanks to this, the glycolysis continues to the ATP with fewer oxygen conditions. Since there is a limited supply of NAD, the electron acceptor must be regenerated to allow ATP production to continue. After the NADH donates its extra electrons to pyruvate molecules, generated NAD. So the lactic acid is formed by the reduction of the fibrate. Fermentation of acetic acid converts fibrate to lactic acid and generates NAD from the NADH.
What are the enzymes? They are organic molecules that can cause chemical reactions that accelerate the reaction rate. Ruba is an important substrate involved in carbon fixation during the Calvin cycle of the dark phase of photosynthesis. The rubis coenzyme catalyzes the incorporation of carbon dioxide into ruba.
The enzyme pyruvate dehydrogenase catalyzes the conversion of pyruvate to acetyl CoA ATP. Acetyl CoA and NADH don't regulate inhibit pyruvate dehydrogenase, while ADP and pyruvate activate it.
The enzyme in lactic fermentation, one is the lactate dehydrogenase. It is involved in energy production that is found in almost all of the body cells, with the highest levels found in the cells of the heart, liver, muscles, kidneys, lungs, and in blood cells. Bacteria also produce ALD. The lactic acid is produced from fibric acid through the enzyme lactate dehydrogenase in fermentation process, and the other enzyme that is part of the lactic fermentation is nicotinamide adenylonucleotide, NAD, which is a coenzyme that is composed of two nucleotides, adenine and nicotinamide, and it is an essential part of the nucleotide that serves as an essential cofactor and substrate for several critical cellular processes involving oxidative phosphorylation and ATP production, DNA repair, etc.
Enzyme of alcoholic fermentation. Zymes, a complex enzymatic mixture secreted by yeast that catalyzes glycolysis or breakdown of glucose and other simple sugars into ethyl alcohol and carbon dioxide during alcoholic fermentation. What are the proteins? Proteins are molecules made of amino acids that are linked by a type of bond known as peptide bonds. They are ordered in arrangement of the amino acids depend on the genetic code of each person. All proteins are made of carbon, hydrogen, oxygen, nitrogen, and most also contain sulfur and phosphorus. Proteins of the photosynthesis. None of the involved in the assembly of proteins required in the first stage of photosynthesis, but a little was known about their role in photosynthesis process.
The research discovered that the CGL71 protein protects oxygen from photosynthesis occurs during its assembly.
Proteins of cellular respiration. Most of the time, amino acids are recycled and used to form new proteins. They are not oxidized for fuel. However, if there are more amino acids that the body needs or if the cells are fasting, some amino acids are broken down for energy purposes by cellular respiration.
Well, this is the comprehension activity presentation, and thank you for your attention, and sorry about the mistakes.